Hello everyone, my name is Abdul-Rahim. I'm a senior student in Alan Wolfe's lab at Loyola University Chicago, and my talk today focuses on expanded culture urotypes analyzed in the context of urinalysis phenotypes through a randomized clinical trial. Here are our affiliations to disclose, as well as our acknowledgement of funding source. As an introduction, standard urine culture was developed to detect E. coli and other fast-growing uropathogens, while expanded quantitative urine culture was designed by our group to detect microbes that may be frequently missed by standard culture. I have a table at the bottom outlining the key differences between standard and expanded culture, including a higher volume of urine, more growth plates, and longer incubation times in expanded culture relative to standard culture. In addition, urinalysis is regularly performed in clinical microbiology settings to assess white blood cell counts, red blood cell counts, leukocyte and nitrite levels, among other measures. The objective of our study was to compare the urinalysis results of women with self-reported UTI across urotypes generated from expanded quantitative urine culture. We hypothesize that urinalysis results will vary relative to urotype. To outline our study design, this is a sub-analysis of a clinical trial of women presenting with self-reported UTI with a total sample size of 225. These women provided a catheterized urine sample and received treatment based on expanded culture or standard culture results. These urine samples were sent to both research and clinical microbiology labs. In the research lab, both expanded and standard culture were performed on all samples, and 4 urotypes were derived from expanded culture results. These were E. coli predominant, gram-negative non-E. coli uropathogen predominant, gram-positive uropathogen predominant, and culture-negative or non-uropathogen predominant. In addition, the clinical microbiology lab performed urinalysis for red blood cell counts, white blood cell counts, nitrite and leukocyte positivity on all samples, which we then compared the results between urotypes. Here is our first table outlining the median red blood cell counts and white blood cell counts across each of the 4 urotypes: E. coli predominant, EC, gram-negative pathogen predominant, GN, gram-positive pathogen, GP, or non-uropathogen predominant or culture negative, NUC, with p-values for the respective comparisons. Our second table outlines the differences across leukocyte and nitrite phenotypes in terms of the relative proportions across each of the 4 urotypes. For example, the leukocyte negative, nitrite negative phenotype corresponded to 57 patients, and we present the analysis across each of the 4 urotypes in terms of relative proportions, with p-values for comparison. To interpret our results, there appear to be specific urinalysis patterns more frequently associated with specific microbiome culture profiles, or urotypes. In addition, white blood cell counts could be used to distinguish between patients that were non-uropathogen predominant or culture negative versus patients that were predominantly colonized by E. coli or another gram-negative uropathogen. Further, there were clear differences in leukocyte and nitrite patterns between non-uropathogen predominant and E. coli predominant or gram-negative pathogen dominant urotypes. Finally, patients in the gram-positive pathogen urotype were distinct from all others in both median white blood cell and red blood cell counts, as well as the relative proportions across leukocyte and nitrite phenotypes. Our conclusions are that there are significant differences in leukocyte nitrite phenotypes and median white blood cell and red blood cell counts that suggest the presence of gram-positive uropathogens in urine that standard urine culture is frequently missing. Additionally, symptomatic patients with low non-zero white blood cell and red blood cell counts and a negative nitrite result likely possess urinary microbes consistently detected by expanded culture but often missed by standard culture. Overall, this supports our hypothesis that urinalysis phenotypes vary relative to expanded culture urotype. Thank you for your time. I would be happy to take any questions.